So you met me yesterday, so here I am again. Yesterday I spoke about the natural history study. Good?
Are you Mike for him? Yeah.
Okay, sorry. He's good. He's the only one who's good. So, and I'm here representing Orphan Technologies. Thank you, Margie, for that great introduction. Again, yesterday we talked, you've seen the slide more times than you probably care for, but in the natural history study, we're confirming what you already know, and that is that all the current treatments do not necessarily lower homocysteine levels as much as the patients and doctors would like. So patients on natural history were complying with a low methionine diet, the vitamin therapy, the betaine, yet in general, on average, homocysteine levels remain pretty high. And so what Dr. Krause initiated and developed for so many years, decades really, was getting to the root cause of the problem, the CBS enzyme itself, which is a tried and true approach for rare disease treatment. So let's see. With that, this is his life's work that I'm about to present. This is what he spent decades, Dr. Krause spent decades working on and really fine-tuning. So our treatment is called OT-58. There's a reason that it's called 58. I'm told there are not 57 previous versions, but close. So he really, that scientific rigor that we heard about is true. I've never, in all my years working in drug development, seen a more thorough approach to drug development. He did everything there could be done. And the important thing to remember about OT-58, one of the many things, is that it's a human CBS enzyme. So it's a naturally occurring, something that your bodies are already making. It has been modified slightly so that it can be given and made easily, but it is human. And I'll show in a slide that won't do any justice to the decades of work from Dr. Krause that all the preclinical, meaning in animals, the work that was done, that it reduces not only plasma homocysteine, but also in tissues. And that it normalizes all those products after homocysteine. And symptoms you can't call it clinical because it's mice, but basically the symptoms in mice are also improved to made as good as wild type, which is the non-models of a disease. So again, this is one slide that represents so much work, but I wanted to, I know that I only have a few minutes, so I wanted to give you an overview. And it's very important to remember, this is in mice, and I always like to say mice and humans are not the same, but it's also very, very, very encouraging of work. So if you're lucky in a rare disease, you have maybe one animal model of a disease. Here we have at least 3. And Dr. Krause's lab used all 3 many, many times. And what he showed was OT-58, that again, plasma and tissues levels of homocysteine decreased and cysteine increased. For eyes, he showed that there was the little fibers that are defective in these mice were restored. Skeleton prevented the osteoporosis in these mice. Even the mice were able to learn better. You put mice in a little maze and they're able to learn better. They also learned that mice on a methionine restricted diet have increased anxiety, which by giving OT-58, that was removed, no more anxiety. And I could go into a lot more detail when there are papers published. His group also, and with us, also showed that OT-58 improved vascular function in these mice. And there are some additional unpublished data yet to come about the diet in these mice and ability of these mice to eat a normal diet. So with that, we partner with Dr. Krause's lab, and really it was the best partnership I've ever experienced between industry and academia. So we worked extremely close. We still work very closely. And essentially, they did all the animal proof of concept work, and we took it on to do the development, the large-scale manufacturing, the safety, and now efficacy that I'll be showing you. So I don't expect you to be able to read this. I don't expect you to learn this, but I want to give you a kind of an overview of the process because it is long and often, you know, it's something that people, it can be frustrating how long it might take. You know, Margie showed years in the making for all this possible treatments. So it all starts with when, you know, in a lab, in somebody's lab, you have an idea. Then you have to, if you're lucky, you have an animal model. In this case, we had 3, at least. And do all sorts of experiments. And then all of this here is really animal work, where you make sure this is a very safe product. Before it ever goes into any person, you have to do quite a bit of very strict testing. You also do the manufacturing. So what was enough for mice is not going to be enough for people. So, and also what was good enough for mice is never good enough for people. So you have to be very strict and follow the rules. And all this takes time. But we've done all that. And for OT-58, we also did what is very useful and very helpful in rare disease, which is a natural history study to understand what's going on. Because again, there's so little out there. And the red arrow, which I can't reach, is where we're at. So phase 1 and phase 2 combined. So what is phase 1? And I'll have another slide showing a little bit more. But phase 1 is where you start in humans and you look to make sure it's safe. So you start often with smaller amounts and you go up in levels. Phase 2 is you keep looking at safety, but you also make sure that it's doing what it's supposed to do. And in our case, we're doing both together. That's what the FDA wanted us to do, and we also did. And we're also manufacturing at an even greater level, and all that is going very well. And so this is where we're at right now. So what does that look like? So we, as Margie mentioned, we have an ongoing phase 1/2 study in 4 clinical sites in the United States, 1 right here in Indianapolis. We also have Philadelphia, Boston, and Denver. And Dr. Krause was able to meet the first Colorado patient who was given this treatment, so I'm very glad that that happened. So there's a very long title for this trial, and I encourage you, if you have questions, not only to go to clinicaltrials.gov, but we also have flyers outside that have that information, and also an email that you can, email, and somebody in our team can respond to all your questions. So, but what does this long title mean? Double blind, meaning that OT does not know what treatment people are getting, And the researchers don't know either, so that no one is influenced in any way. Randomized means that you get either, most likely you will get drug if you join this trial, but there's also a placebo. Why placebo? It's so that you can compare the drug against no drug. And it's the highest level of scrutiny that you can do for a clinical trial. And, but again, most people are on drug. So it's a phase 1/2 study, as I mentioned, and it's to look at safety, tolerability, meaning how does it behave in people, how safe it is in people. Pharmacokinetics is just a fancy word to say, you know, how quickly do blood levels rise of OT-58, and then what does that look like? They go up, and then they go down, and then you give drug again, and what does that look like? And also pharmacodynamics, another fancy word to say, do your homocysteine levels go down? Cystathionine, does that go up? And many other metabolites. And then clinical outcomes, that's a part of a phase 2. So let's look at what's happening elsewhere. Let's look at the, blood vessels, eyes, skeleton, brain, et cetera, all the different systems that are affected. So again, if you have questions, we have an email that you can easily ask about. You can go to clinicaltrials.gov and look. So where are we right now? Again, same graph as before, but this is a little more blown up. So the natural history is sometimes called phase 0, no treatment. That's ongoing. Phase 1, phase 2, that's ongoing. And we are getting ready for talking to regulators, the FDA, and move on to the next phase, whether it's, typically phase 3 is the next step, but again, there are many ways to go about, and there'll be lots of discussions with FDA to find out what that looks like. And finally, I'd like to encourage you, as I did yesterday, to be part of a process and whatever you can do. And there will be, as all these treatments progress, FDA and others in regulators will want to know what you think and what you want. So I encourage all of you to be active in that. And I think that's about my 10 minutes, but I don't know if you want me to answer questions.
We'll have questions for the three of us.
And finally, not the most important, thank you for everybody who contributed their time to participate in these trials. And the families, I know I can only imagine how much effort it takes. And we tried to make it as easy as possible on you, but we really appreciate, and we could not succeed without you. And with all of you, thank you so much. And thank you. I see you in that work.
So we need you to come and transfer that.
Here, if you hold that, it's...
Position that.
I'm the director of Biology and Toxicology.
So I'm now pleased to introduce a researcher from Aeglea, Chris Dage, who's the director of toxicology and is it pharmacology?
And biology.
And biology, close. So he's going to give you an update on the compound that I discussed that Aeglea was putting into research.
Okay, well, good morning, everybody. Can everybody hear me okay? Good. Thanks so much for having us out, Margie and Danae. Really appreciate it. And I know all my colleagues, there's 3 of us here, all have really found this experience to be very invaluable so far. My name's Chris Dage. I'm the director of biology and toxicology at not so easy to pronounce company name, Aeglea. We are a biotherapeutics company, and our goal is to develop and bring to patients next generation human enzymes. We were actually spun out of George Georgiou's lab at the University of Texas in Austin. and we have our headquarters remaining there to this day. I wanted to give you a picture showing you what our team looks like, and I realized as I was doing it, I don't even know where I am in it. Turned into a bit of a Where's Waldo situation, but sure enough, there I am. There's the top of my head, maybe my ears, they're in there. So just to let you know what we're working on in general, we have a number of different research programs that we're trying to advance to bring therapies to patients. We're targeting a number of different cancer types, but our most advanced program is actually an arginine degrading enzyme for the treatment of arginase 1 deficiency. And that's actually all the way out into phase 3 clinical trials right now. But what I'm here to talk about is our AEB4104 program, which is our homocystinuria research program, and where we're at, and just give you an update. So how does our technology work? What are we trying to do here? How are we different? What we're doing is taking already existing human enzymes and mutating them to have different activity. And so what makes ours unique right now is that we're using an enzyme that's actually just downstream of where CBS breaks down homocysteine. So we're using the enzyme that breaks down cystathionine, so cystathionine gamma lyase. And what we found is that that actually has a unique ability to modulate activity of a number of different metabolites, one of which is homocysteine. And so what we're trying to do is to dial out the cystathionine degrading activity and dial way up the homocysteinase activity. And so it's taken many, many years to do this, of course, as everybody knows in the research world, but what we've come to is ACN177, which is our lead homocysteinase molecule. And I know this is kind of painful, showing the dreaded biochemical pathway slide again, but I'm just using it to show in context where ACN177 fits in. So as we all know, mutations to CBS enzyme lead to the buildup of homocysteine in cells, and later it spills out into the blood. And so that's where we can kind of come in here. So it exists in 3 different forms. You have your protein-bound homocysteine, as was mentioned yesterday, and then also you have homocysteine and homocysteine, the dimerized form of homocysteine. And so what we found, and what makes ACN177 unique, is that it actually directly and rapidly will degrade both homocysteine and homocysteine and produce the all-natural byproducts that you would see with CGL. And also we see these levels stay down for quite a long time. This is where we're at with developing the molecule, and I just have a brief summary slide to wrap up to show you where we are in the whole development process. So we've identified our lead therapeutic, check, ACN177, and we've done years of proof of concept work, looking at a number of different animal models, understanding how it works relative to the disease, and if we have a potential to have a therapeutic moving forward. We've been very satisfied with the results, and so we are moving forward with this. And as was just mentioned, there's a number of things that have to happen before you can actually advance into clinical trials. Well, first you have to like the drug that you have and think that it has a good chance, but then you have to figure out how to make it on really high scale levels, which isn't always straightforward. So basically that manufacturing process, you need to have enough of the drug around to support toxicology studies where we have to make sure the drug is safe before we go into people, and then also into the phase 1 clinical trials where we're finally testing in patients and people. And so where we're at right now is we're actually identified our manufacturing process. That looks good. We're moving forward with that, and we're actually just about complete with our toxicology studies, which will enable us to go into clinical trials. At the very end of this year, that's the plan, that we should be ready to file some sort of regulatory initiation. And we're actually hoping to initiate the clinical trial for homocystinuria second quarter of 2020. So coming up very soon. That's all I have. Thank you.
So maybe I will just hold this microphone and ask Marcia to come up as well and Chris to stay. And we'll open up for questions. We have about 10 minutes per the agenda. And who's ever going to answer the question, I will give the microphone to. Please repeat the question for the purpose of the video and give your answers. So I saw the first hand up, Rose.
Yes, with regards to the recent presentation at AEB4104, when you spoke about utilizing the CDS downside enzyme, I don't know the technical term there, is that said to be within the patient itself, or utilizing like outside the body enzymes from different, you know what I'm saying?
So you're asking whether or not CGL is, we're initiating the production of it in the body, or if we're introducing it as the latter. We're making us out of the body CGL, the modified CGL, ACM177, and using that, injecting that to treat the disease.
And where's it coming from, your initial CGL?
The initial CGL we manufacture in the laboratory.
So it's synthetic,
Yeah.
but it's mimicking.
And it is a biologic, to answer your question. Yeah.
Once you said injecting, I knew it
Yeah.
was. Thank you. He said it is a biologic because it will be injected. That's a usual clue. Yes, right here.
So what's the difference between that and cystidine?
So the question I'll have Chris answer is, what's the difference between the Aeglea compound and cystidine?
So the Aeglea compound is a totally different enzyme. That's the difference. We're actually taking, so it naturally occurs in the body to degrade cystathionine. And so what we've noticed is that we can actually modify it just to focus in on the homocysteine levels.
Yeah.
Sorry, I thought you were
Going to
going.
one of my slides to talk about cystidine. I figured you probably didn't want to do that.
Thank you.
Probably a helpful slide to have up for the question.
Yeah.
Okay, so cystidine is a form of betaine, which is up here at the top of the cycle, and Kim can add on if I get this wrong. But betaine helps to convert homocysteine back to methionine. So you're up in the top half of this. You are accumulating homocysteine because you've got this blockage or the dam forming here. And so high levels of homocysteine cause damage in the body. So betaine helps to convert it back to methionine. So it all stays up here. You can't come through this dam. I'm assuming that the Aeglea compound, as it metabolizes homocysteine, you're then able to come down this pathway and make some of these other compounds, theoretically?
So it'll make the natural byproducts that you'd see with CGL breaking down cystathionine.
So it comes down.
Yeah. Yes.
Other questions? Mel?
Could you help me understand that cystathionine is degraded into, what is your technology again? I mean, what is the pathway again? Could you explain that one more time?
Yeah, so the tricky part here is that it's really kind of an alternative pathway. So we're able to, instead of utilizing CBS, right? So we're engineering the enzyme that does this process right here and getting it to kind of come back through here and break down the homocysteine directly. So it's a naturally occurring enzyme, but it's really a modified CGL, which is cystathionine gamma lyase. Yeah.
Thank you. Oops, sorry. Yes, Ben.
You mentioned it's an injection, would be like 2 times a day or something that you would inject? Like how would it?
So the question is, how many times a day or a week or month would the injection occur?
So that's what we're trying to figure out right now. I mean, obviously we want to minimize that as much as possible. We are looking into different ways of injecting subcutaneous routes of delivery and so forth as well. But we want to make that as burden-free as possible.
What about the OT-58?
Yep. Same question for OT-58. How often would it be administered?
So it is being administered right now, weekly, and part of a phase 1/2 process is to optimize and see what makes sense, not only from a scientific point of view, but also from a ease of use. So it's sub-Q, it's, if you think about, maybe the closest one would be, although not as frequently as like insulin, We intend to have this be self-administered and just as easy as can be for home use eventually. So we're looking, part of a phase 1/2 is you figure out what makes sense, and but that's the base case.
And when she says self-administered, obviously, for the parents in the room of HCU patients, young patients, it would be parental, or the parent, we hope, would be administering that. Yes?
This is for Orphan Technologies. You touched on this a little bit, but when can we expect to see results from the human study, as you've already done with the murine study, as far as clinical sort of improvements, eyes, vascular, et cetera?
So the question is, when can the community expect to see results from Orphan Technologies' phase 1/2 trial of OT-58?
So again, as I mentioned, it's a double blind. So we are able to see data right now, but we don't know who it belongs to, right? So, but as far as being able to talk about it, we expect sometime in the first half of next year to be able to share something. And again, as the way it works is that you can share some, and then as you finalize everything and kind of put the final touch on the final report, then you can share more. So we'll be sharing as much as we can, as soon as we can, but pretty soon. Mm-hmm.
And usually scientific data are presented at scientific conferences. And so I know I personally, on behalf of HCU Network America, am encouraging Marcia to try to get it to the metabolic clinicians meeting, which is late April of next year. And so we're going to see, if we can put some subtle pressure into the system, because they've got rules about when your abstracts have to be in by. They want to have all the data by then. But Kim, maybe you can help us. Can abstracts have X's that you can fill in later? We're going to see what we can do, but we know the community is anxious, and we'll do everything we can. Mel?
One question for me, for both of you actually. Is there like for treatment for diabetes, you have like multiple medications that you take? Would like both or like multiple therapies can be administered for the same person?
So the question from Mel is, could these two compounds or multiple compounds be administered for the same person? I mean, I'll take it first. It's in general, as a pharmacist, I feel very qualified to answer this question. Certainly for any disease, if there are multiple products available, you can use them in combination. And so, for example, the one research program I talked about that Ken McGlynn has, you would still use betaine, but if you added formate to it, theoretically, based upon his animal data, he thinks that would make betaine much more effective. And so potentially that may be all you need. Could you potentially even avoid diet? Studies will tell us. So for any disease areas, you could act at multiple points along the continuum. I'll give you my personal point of view. Biologic products are usually not inexpensive, so if you can get by with one, very, very complicated to produce and manufacture, et cetera. So, ideally you would want one of a very effective product, but certainly there are opportunities to use things in combination if it makes sense. Usually you don't have two things that kind of work at the same point in the pathway. More likely,
the same point, but you also cannot do trials in which you're on more than one trial drug at a time, depending on the rules of the trial,
Right.
will depend on what your other therapies are allowed to be. But right now, we use multiple therapies for every patient anyways.
so what Kim said is multiple therapies are often used for patients, but in terms of a drug trial, it would be very difficult to do. And the FDA actually likes you to first get approval of the independent products, and then down the road, test them together.
Not necessarily
But
during the trial, I'm just saying.
not necessarily during the trial, but clinicians can do whatever they want. I mean, they of course do what is in the patient's best interest, but if both of these products were approved, and Dr. Chapman thought using a little bit of each of them as opposed to a lot of one of them was better for her patient, she could certainly do that.
So to add to this, in other rare diseases I've been involved in, have been two of the same type of enzyme therapies, Fabre and Gaucher, two that come to mind. And some work for some patients and some work best for other patients. In this case, though, I just want to make sure that everybody understands that in our trial, people can still be on betaine, vitamins, et cetera. And the idea then is it'll be, whatever works for the patient, you can start to play with doses of various things, of these current treatments, as well as of an enzyme and obtain and get the best care that you can get, and working closely with your physician, of course. So, but yeah.
Come down here to see if this is kind of fun, having two competing companies. So.
Not a whole lot to add to that, actually. I think without giving away too much of our strategy going forth, we do anticipate running into that people want to stay on their existing therapies, and we want to make sure that ours works well with existing therapies and then also hopefully independently.
And if I can add. Actually, it's, yeah. No, it's, I've been doing this for a while, and my personal feeling is the more, the better. Patients need choices. So let's do enzyme, let's do gene therapy, let's do everything, let's do chaperones. Whatever you can do. And not everything's going to work for everybody. The more choices you have, the better off everybody is. So.
Amen. Amen. Yes, Rose?
With regards to OT-58 and the possibility or preliminary data being kind of revealed in the early part of 2020,
Yep.
With completion of phase 2, what is the anticipation, or is there a forward plan already set based on some of the data that you're?
Yes.
So the question is, assuming we get data revealed in the first half of 2020, is there a plan to move forward? And Marcia will give us hopefully a sense of what that is.
To the extent I can say, yes, obviously, we do have a plan. A lot of it depends, in any clinical trial for anything, depends on the data that you get. So once we have all that data in a format that we can go to the FDA and talk to them, they're already, FDA really wants to help new treatments come to fruition for rare diseases or for any disease, but they're really working with us. It's not a, us against them at all. So it's a discussion. It really is a discussion. You propose things, and they really understand that patients need this sooner than later. And so they work with industry to make that happen. So I can't give you a definitive answer because I don't know that answer. But we're doing the best, and yes, plans. I don't know if you noticed, last night I left early and I went back to my room to work. So we are working really hard. Yeah.
And certainly one of the things we intend to do through HCU Network America is keep the community aware of any of these programs that are going forward into human trials. We heard there may be a phase 1 in 2020. There could be a phase 3 in 2020. You would expect that, there'll be different sites around the country, maybe even Europe, and we will make you aware and help as much as we can for those interested in participating. Danae?
We've been encouraged as an organization to start speaking to the FDA in advance to kind of help liaison and get things moving.
All right, I'll repeat what Danae said, and we have been encouraged as an organization to liaise directly with the FDA to, let them know how patients feel about the current approaches. And, one of the important things here is that we want to make sure they thoroughly understand that, yes, while you can manage your homocysteine levels with diet, that is not a situation that is optimal or ideal for the patients, and that new therapies would make a huge impact on quality of life from that perspective. Mark?
Marcia, to just kind of go back to that question earlier, which is great.
Yes.
You're going to approach the FDA soon, you're going to have data, but the natural history study is really going to be a huge lever for you
absolutely.
to convince the FDA of the patient population, the need, and then obviously the data just really kind of makes that conversation with the FDA a bit more impactful, right?
So I'll just repeat that and then let Marcia comment. Mark was saying that natural history study is going to be of major benefit as the company is talking to the FDA and will certainly be helpful in your discussions. Marcia?
Yes, and again, I want to emphasize how valuable your efforts are in the natural history study, because it is truly a foundation for the entire field, and all the other, EHOT and every other bit of data we can get. But yes, in rare disease especially, having a natural history study, it makes a very big difference. And we are analyzing, as I showed some analysis yesterday, but we continue to analyze, and we'll be publishing that data. We want to share with the community and obviously with FDA as well. So it's an education for everyone. We're learning, FDA is learning, everybody's learning, and you're teaching us, so thank you.
Yeah, and the other aspect I'd like to comment on, what excites me also about the natural history study, I think it's going to be very helpful in reimbursement discussions. Having come from a world of pharmacy, but then also having been very involved in getting reimbursement, Merck had a portfolio of about 25 different products. Every time we introduced a new product, we had to convince payers in the U.S., health management organizations, the government, et cetera, to provide those products to members of their plans. And I can tell you that the data that shows that current dietary management is not working, and as sad as it is to see on the plot that Marcia showed, that some people are still at homocysteine levels of 150, 200, 300, 350. While that is very sad to see, and I feel for those patients and hope they can get it down, it does convince those who are making the decisions about whether to provide reimbursement that this is a very needed therapy, and you shouldn't look at it as, oh, well, those patients can just control their diet. Why should I be willing to reimburse for this new product?
That was one of the biggest struggles with PKU.
Yeah, and Dr. Chapman added in, that was a huge struggle with PKU drugs that payers said, well, gee, you can control it with diet. So hopefully their work on getting reimbursement for their recent introduction, which is kind of, it's an ERT type product, right? Pegvaliase, is it? Pegvaliase came out last year for PKU, so hopefully that paved the way. Yes?
About the natural history studies, do they get shared? If it's the foundation for what you're doing at Orphan Technology Center, is Aeglea, is that just shared with all the different companies working on?
So the question to Marcia, put her in the hot seat, is the natural history study is great information. Do you then openly share that with all the other companies in the field?
So the answer is yes, in the sense that we're publishing. Yes.
I'm gonna answer part of this
All
too,
right,
because
okay.
Take it away, Kim.
the National Organization of Rare Disease announced approximately 2 weeks ago, maybe 3 weeks ago, of a mechanism called I Am Rare. And I Am Rare is a database stored out of someplace in Arizona. I can give you guys the contact people, which will summarize all the data that is gathered by hopefully all the pharmaceutical companies, all the natural history studies. It's going to hopefully reflect the same data sets as what's seen in EHOD. It is funded by the FDA in terms of, and NORD, in terms of recognizing that natural history studies are really significant. And this is a hope that different studies can put all of their data, including historical data, into the same database. It clearly will not designate where each piece came from. It's a little different than a natural history study that follows an individual across time. This is a compilation. It's all together. And the goal is so that data is freely available to researchers and companies as they approach the FDA and try to get approval for their drugs.
Thank you, Dr. Chapman. Any other questions? Well, great. Well, we covered a lot of information. I really give all of you credit. Some of it was pretty technical. I think all of us tried to present it in a way that, you're all obviously a super informed group because of your connection to HCU, but I really am glad to see your interest and excitement, and I hope you agree that all of the work these companies are doing, the researchers are doing, gives us greater hope for the future. So thank you to our presenters for sharing their information and for your work.